Are you working on prebiotic, probiotic, or postbiotics, sir? In your
No,
research
no, no.
interest?
I
Mhm.
only do zoology teaching.
Geology teaching. It's a great, sir.
Yeah.
Now, the Urvasi Anand.
Good evening, sir.
Good evening.
Sir, I am working as a PhD scholar in CSIR CFTRI Dr. Syed Mustafa Sir's lab.
Okay. Thank you, Urvasi. So, shall we start now, sir? Session.
Yes, of course.
Yeah, because 13 have joined, so you can start our session now.
Urvashi, you are doing PhD or?
Yes, sir, I am first year student.
I have taken your introductory class during
No, sir.
orientation.
Orientation, yes, sir.
Oh, okay, okay.
So, good evening, everyone, and I welcome all of you in this today's roundtable discussion. The topic do in postbiotic will be a new therapeutic window. So we all are familiar with quite familiar with the microbiome based intervention that the starting from the probiotics, prebiotics, and then to the synbiotics. And the use of all these has contributed significantly to our understanding that how the gut microbiota influence the human health. So today we would like to bring our attention to the relatively the newer and the rapidly emerging concept that is the postbiotics, and to explore or to discuss whether the postbiotic truly offer a better therapeutic window or not. So let me begin because this in the last session we have already discussed on the what the postbiotics and what how these are better than the probiotics and the prebiotics. Okay, but today I would like to put those three questions because Dr. Prakash Alami sir and the others expertise are here that means can we fully replicate the postbiotic means adaptic interaction to live microbiota how and how can we define that the optimal dosing or the combinations or the long term effects of the postbiotics, okay, or that how this will work in from the means problem can be arise for the microbial colonization or the competition with the gut ecosystem or not. Okay, so I would like to welcome all in this roundtable discussion, and I request all of you to or I expect the active participation from all of you. So I would like to open this platform for the discussion now with these three questions. Okay, and one more question with me that I will ask Halami sir at the end of the session.
Yes,
So,
of
sir,
course.
today we would like to start from you, sir.
From me?
Yes, sir, from you, sir. Because
Wonderful. Thank you. So once we say the therapeutic, once we say the postbiotic, we have to actually differentiate between the probiotics, and then we have seen that probiotics has few advantages, disadvantages, I mean, where we have to have the live bacteria. Then there are the number game, that live bacteria should be in sufficient number, and then storage stability, other issue. The other inconvenience which we see is the antibiotic resistance that can be transferred, right? And then of course some probiotics can be actually infection, can actually induce the infection also, right? So infectivity is there with some of this probiotic, especially in a small children as well as the immunocompromised patients, pregnant women, right? Now, as far as the disease prevention is concerned, probiotics to some extent, yes, but the therapeutic application is concerned, postbiotic molecules, the extract or the dead cells, formulation of the dead cells can actually work much in a better or effective way, and also can be a very promising with reference to the treatment is concerned, most of this lifestyle diseases, in other words. And as I told the last time, that the presently the postbiotics we are actually using for the two at least the diseases. One is the diabetics, and another is hyperlipidemia. And we could actually have our very good lead available. That student, she didn't join today online. um, with reference to both these diseases. And we could see that very promising results compared to the standard drug. All right. Just I wanted to inform all the audience that we have actually the edited book on the postbiotic health and industry, and we have almost 15 chapters in that. So first section is actually fundamentals of postbiotic. Those um, participants would like to have any specific chapter, they can always ask me for searching this, rather than searching the literature here, there. Okay. So first actually section talks about the um, 10th chapter comprises of the fundamentals of postbiotic, where we actually ask the researcher from the different organization to give a book chapter on the postbiotic and host for probiotic microorganisms, the postbiotic lactobacilli, postbiotic bifidobacteria, postbiotic from lactococcus, then from Saccharomyces, then oral microbiome and human health, separation characterization of a postbiotic, genomic characterization of the postbiotics, and unlocking the health potential of the postbiotic. I will directly go to section 3, which actually talks with reference to application of postbiotic in health and pharmaceutical industry, where we actually have a chapters comprising of postbiotic, a health promoter in human, then integrative multiomics, understanding postbiotics and their role in human physiology. Then we know that several of these antimicrobial compounds are also considered as a postbiotic. So we have antimicrobial effects of a postbiotic. Then prevention of microbial infection by postbiotic, prevention of food allergy using a postbiotic. Even we have the chapter on this one. And even right now we are actually working on the probiotics with a anti-allergenic effect. We have actually this going to start a new collaboration with some allergic organization, anti-allergic organization, I mean. And then anti-cancer and antioxidant activity also of this postbiotic has been reported. Then anti-inflammatory effect of the postbiotic. Then we have actually the anti-atherosclerotic effect of the postbiotic. So several such diseases are covered, literatures are available, and even some of this formulation, which I believe I told during last roundtable discussion, that some of this formulation those are available in US and Europe commercially, for few of these diseases was described last time. So such vast information is coming up, and recently International Life Science Institute, ILC India, in fact, asked me to compile a status book on the postbiotic application. So that one actually right now is under publication from our side, and then of course we are actually preparing a book chapter on a postbiotic from the bacterial sources. So, and as I told already that we have actually the DRDO ongoing project on the postbiotic, where we are exclusively working on using the two bacteria. One is the bifidobacteria and another is the lactobacillus, and looking to combat the two diseases. One is the diabetics and another is hyperlipidemia. So the postbiotic has been characterized, preclinical studies has been undertaken, and then this material, the postbiotic which has been fractionated and prepared, will be incorporated and used as a functional food. So this is what actually the present status from our side. In a gut microbiome of 2022 issue, there is actually a very interesting article by Colin Hill, who works on the lactic acid bacteria. They knows about the Colin Hill. The clinical evidence for postbiotic as a microbial therapeutics. So I think this article fits very well with reference to the today's topic of discussion. So everyone actually can go through this article, or those who are interested on this, which actually describe the mechanism, postbiotic mechanism of action. Mostly you will see it is the immunomodulation system, right? And then uh, several such informations are available now on this postbiotic. Of course, there are some of this concerned also using this postbiotic, but then since these are mostly of the food grade organisms, we should not have much difficulty to explore them. Maybe perhaps from the next generation probiotics. In fact, just last week I had a lecture on the next generation probiotics, where we use several of these gram-negative bacteria, gram-negative bacteria. And live gram-negative bacteria cannot be used as such as a probiotics. So in such cases, we have to use them as a source of biotherapeutics or the postbiotic molecule. Okay. Okay, that's all from my side. Now
Yes.
we can.
Yes, sir. Thank you so much, sir. So now we'd like to take some questions. If you have any query or your questions to the Prakash Salami, sir, you can unmute yourself or you can write in the chat box.
Exactly.
Am I audible to Prakash Almani, sir?
Yes, sir. You are audible, sir. Good evening, sir.
Sir, yeah, I was just listening to the sir's speech. It was excellently drafted and well presented.
Thank you,
Sir,
sir.
I want to know how probiotic help in diabetes.
Probiotics we have used for the diabetics treatment. We induce the diabetics, and then we could see, especially with reference to GLP-1,
Can you
glucose
can
metabolism.
you kindly elucidate, sir?
Yeah, we have the preclinical trial carried out for the diabetics, and we have actually
Yeah.
seen the markers beside the pro-inflammatory and anti-inflammatory cytokine level. We have seen the GLP-1 level.
Okay.
Yes. Well-established study, and we have actually identified culture lactobacillus fermentum, our own isolate. And
Okay.
2 years back, this publication is there. In fact, I wanted to go for a clinical trial. Madras Diabetic Association, I discussed with them, but the quote which they gave was quite expensive, somewhere 12 to 15 lakhs.
Oh.
Yeah. Because they have to spend.
Okay. Thank you, sir. Yeah, thank you. Thank you, sir.
Thank you.
So if anyone want to share your research insights or any knowledge on this postbiotic, please
But now,
unmute.
just a minute, just to tell Yashwant sir that with reference to the postbiotic, actually, for the diabetics,
Sir.
actually very good results.
Mhm.
That work is in progress, and we have already completed the preclinical trial. We are preparing that manuscript because
Sir, may
this is actually the
I
project.
kindly know how the study was carried out, sir?
Yeah, please go
How
ahead.
the study was carried out?
Yeah,
Am I audible, sir?
we induced the diabetics, and then diabetics
Sir.
induced rats were actually fed with the postbiotic molecule, postbiotic
Okay.
preparation. And then we check the gene expression. Then we have the gut microbiota analysis also. And then the some of this vital organs tissue, especially the pancreas in case of the diabetics. And then we could actually see the severity of the disease actually reduces.
Oh,
Yes. I hope once this
because every third person.
work gets published, it will
Sorry, sir.
be interesting to share right now, because
Yes,
this is actually the DRDO project. And actually the
sir.
story is something like this, because the soldiers which are actually deployed at higher altitude, they usually
Yeah.
have this problem of either diabetics or hyperlipidemia, beside dysbiotic gut. And they actually just come
Mm.
back, within a 15-20 days or within a 1 month of time. So they will not be actually able to withstand the higher altitude condition.
झालं येतो म्हणे
And,
निघाली तिथून
and that's why actually
येतो म्हणे
DRDO actually is interested to find some through the food source if this can be prevented or cured to some extent. Exactly.
Thank you, sir. Thank you very much.
Thank you.
So any more question or anyone would request that anyone can unmute and can share your research insights or the any information on the postbiotics. So Dr. Anita Tilwari, Dr. Madhavi Rane.
So has the research only been done on the rats or any other organism has also been used?
Now, literature shows that there are several clinical trials also being published. That
Okay.
includes for the diarrhea. The
Okay.
diarrhea, most of this antibiotics is a chronic diarrhea, irritable bowel syndrome,
Okay.
Helicobacter pylori infection, where the clinical trials has been established and has those data has been published already. What I was referring to the preclinical data, as you said, rat model, because as a functional food, preclinical data is more than enough. Because we treat it as a medicine at this moment, we are actually considering as a food component, food material. So we know that any metabolites produced from this grass organism also is considered as a grass status, generally regarded as safe. So that can be actually incorporated and included. And both this culture which I was referring to, these are our own isolate, and this bacterial species are reported in a FSSI brochure also, that these cultures can be used as a probiotics. Yes. So we are
Thank
getting
you.
the metabolites from these cultures.
Okay.
Sir, one question is there from
Uh-huh.
the Sayuri Singho. Since postbiotic do not contain live bacteria,
Yes,
they cannot colonize our gut or continue replication like probiotic bacteria can do.
exactly.
Just 1 minute.
Yeah, because to
So
have
are
the
not their effects more temporary compared to the probiotic?
no, actually probiotics also we are supposed to consume regularly, daily. Not all the probiotics will colonize, they also get washed out.
Mhm.
Yes. So
But
even
if you compare the postbiotic and probiotics, which one will be the longer effective, sir?
no, since both are actually the no, one is the live bacteria, another is the metabolites.
Mm-hmm.
And no, as far as the industrial application perspective, postbiotic. But from the health perspective, but then as we seen initially, that probiotics also has one or other disadvantages, especially the viability is concerned, with reference to the infectivity is concerned, with reference to their gene transfer is concerned. Now, biosafety assessment of the probiotics is actually mandatory, because some of these probiotics will have virulent genes, hemolytic genes, lecithinase genes, more importantly, the antibiotic resistance. So that has to be transmissible antibiotic resistance. I think you must be actually dealing this topic in your other activities also. So if it is a transmissible antibiotic resistant genes, we have to actually avoid those probiotics to be incorporated in the food. So in such cases, we just take the active fraction, where the dosage is confirmed or standardized, and then directly you see, and then no side effect, more importantly.
Okay.
Yes. Both are
Any
growing.
more?
Both are the
Mhm.
growing.
Sir, one more question from the Dr. Madhavi Rane. AP core. I don't know about this. 500 mg is good for diabetic people. It
What
is
is
in the
that?
chat box. EPIC or AP core.
Epicor 5 is good for diabetic people. Epicor is what a standard drug?
Also, I don't have idea about it. Dr. Madhvi, can you explain this one question?
Sir, this is available in the market.
Epicor, yeah. Epicor postbiotic. Yes.
Yeah.
I have heard. We are aware about this.
So is this
Derived from
good?
fermented Saccharomyces bacteria that support the immune and gut health. Yes. Tell me.
Is this good for the diabetic people?
See,
Consumption of this tablet?
basically, postbiotic are doing only two things. One is immune response induction, where the cytokines will be produced, which actually pro-inflammatory will get suppressed and anti-inflammatory will get induced. And similarly, the gut homeostasis. All right. So this is what actually the gut microbiome stability, the richness in a useful microflora. There will be a high diversity and then richness of the useful microflora when we take this postbiotic or the probiotics also. Right. So this is what actually the information is with reference to the postbiotic, and then as such Saccharomyces boulardii, most of these people must be aware. Antibiotic associated diarrhea is being treated using the Saccharomyces boulardii. So if it is in the dead form also, we are just consuming and it is useful. All these days we were not aware, right? So several such fermented food also are the fermented and then fried or dried or cooked. Also will have the postbiotic, in other words. I hope it is clear to you. So similar to Epicor, there are
Yes,
several
sir. Thank you so
formulations are
much.
available actually. Some of these companies
Yeah.
are now actually marketing.
Yes, sir.
Yes.
Thank you, sir. Thank you, sir.
Yeah.
Excuse me, sir. I have a small question about the stability of these probiotics when they are made. And I have a genetic engineering background that was very long back. And I'm not very sure if genetically modified organisms can be used just like how we use it for production of insulin. So this method can be very much effective to produce a large scale. So is that thing going on already, or is it just on the charts, sir?
Genetically engineered probiotics are also considered as a next generation probiotics, but they cannot be considered. They cannot be explored for the consumption because it will be a GM are genetically modified organisms. And genetically modified organisms, one of this reason is the antibiotic resistant genes or the foreign genes. So we don't for the experiment purpose, for studying the mechanism, yes. But as far as the consumption is concerned, right now our regulations do not permit.
Okay.
Right?
Okay.
Yes.
Yes,
Some specific metabolites can be produced, in a food grade organisms, and then that can go as a biotherapeutics.
Right.
Exactly. Or now the genome editing is coming up, right?
Yeah.
So genome editing is more a natural engineering, which is being actually very much proven in the plant biotechnology. So if it is that, the researcher has to come up and then see how best it can be explore that area also.
Yes.
So can you use that genetic modified organism for the postbiotics?
Yeah, that is what actually, because once you do genetic modification, just like the GM crop is there, GM plant is there. So we don't bring it in the consumption as a food. What we have now genetically modified is
so not means genetically modified means you can mutation in some genes so that they can produce a large amount of the metabolites. Is this possible?
Yeah, they are made to produce these. Suppose we use yeast, safe culture of yeast, and then we harvest only the metabolites and then purify them. Is it going to be a future thing, sir?
No. Yeah,
no, it
not
won't be
for a
still
consumption
accepted.
purpose. Because
All
it will be considered as the GM food.
Okay. All right. Okay.
What we have now genetically modified only the cotton
Okay.
and tobacco,
Yes.
Yes.
Sir.
Of
Yes.
course, what we get in India, the soya from US is a
Yeah.
genetic, or corn, they are actually genetically modified, but no
Yeah, and
one,
apples too. Yeah.
or even papaya,
Yeah. Many. Yes. Yes.
So
Banana.
some of these fruits are genetically modified, but we are just consuming because these are not produced in our Indian field, you know.
Okay, sir. And another doubt was about the stability of these metabolites after being produced. So if we harvest them and then store it, what would be
Yes.
the right additive to be increasing the shelf life of these?
Yeah,
Uh,
once we partially characterize, then no, freeze drying and then keeping in a dry powder form will not be that difficult compared to the probiotics. Actually, in probiotics, viability is the major issue.
yes,
And
sir.
within that, to some extent, you can are okay with the lactobacilli, but with bifidobacteria, very, very difficult.
So,
And then when we consume also the bifidobacteria, they actually they reaches to the target site is only 1 or 2%. That means 98, 99% bifidobacteria will be actually dead during the transit.
yeah.
So this is actually the situation scenario.
Okay, sir.
Supplementing this bifidobacteria in a live form in a desirable number. What we have regulation now in the product serving size, we should have 10 raised to power 7. How much they reaches, we never knows. And that's why this prebiotic concept is very much valid, because prebiotics helps this bacteria to grow in abundance, in more
Yes.
number. Right.
And also in spore forms, right?
Another one is the spore form like bacillus, but then most of these bacillus has the virulency and
Okay.
toxin production abilities.
Yes.
We have actually a few of these bacillus cultures also characterized, and they are also producing the spores. But then they are not that normal microflora of our GI tract like bifidobacteria and lactobacillus. Once we say lactobacillus, small intestine, bifidobacteria, colon, large intestine, everything is done. We will be having several hundred thousands other microorganisms, but these two bacteria actually determines the gut homeostasis or the balance between the good and bad bacteria. If we consume these two bacteria daily, we are healthy. We are free from diseases. We are free from any allergic reaction. We are free from the dysbiotic gut. We are free from the bowel movement, inability to digest the food, stomach ache, in other words.
Yes. So that gives me another question. For people with lactate
Lactose
allergy and all the
intolerance.
soy allergy, will
Yes.
this be an answer for those people?
Lactobacillus is most
Yeah,
of the lactic acid bacteria, they hydrolyze the lactose.
yeah.
They will have the beta galactosidase gene. Invariably, lactic acid bacteria, why they grow? Because of that issue only. The lactose, beta galactosidase production, because they have the beta galactosidase gene, and then it takes care of the hydrolyzing the lactose. And this is how actually the milk is being hydrolyzed or curdled, because most of the lactic acid bacteria, they grows in the milk as a substrate, because the sugar is the lactose, right?
Yes, sir. Yeah.
So that is actually taken care, in other words.
May I come in, Prakash?
Yes, sir, of course.
Sir, there is one question I want to ask you regarding hyperlipidemia.
Yes, sir.
Nowadays, majority of people are advised to take hyperlipidemic drugs, though the normal levels of lipids are the levels of lipids are normal also.
Yes.
Okay. And my question is, sir, how the probiotics will help this controlling hyperlipidemia?
Cholesterol reduction, metabolism.
Pardon, sir?
Cholesterol metabolism.
Cholesterol. How actually it works, sir?
Cholesterol metabolism, we actually have three different pathways. One is the absorption in the cell, then degradation of this cholesterol. One of this lactic acid bacteria do degrade this cholesterol. And what is the bile conjugation, I believe, one more with the bile. So bile salt also will take care of the cholesterol metabolism. So we have actually in one of this paper where we have seen this property, what exactly you asked, for bacillus we have proven.
For bacillus.
Yes, Bacillus licheniformis. And a very convincing results with reference to the cholesterol metabolism. Now here, as you said, we are actually looking for the hyperlipidemia using the postbiotic.
Postbiotics. Okay, sir. Thank you.
Thank you. Thank you.
So, thank you. So now,
Specific information I am unable to get suddenly because this DRDO project actually is under progress. And then that bacillus work somewhere 15 years back we carried out, 2011. Bacillus utilizing this cholesterol or associated with the cholesterol metabolism was done somewhere in our laboratory in 2011. This
thank
other
you, sir.
people also can interact.
Yes, sir. That's why I would like to request now Tejaswani,
Yeah.
Anjali, and
And
Sahana
Anjali
to.
also, because Anjali also is now working on some of this postbiotic,
So with Anjali, there are two questions here, now. The first question I think we have already discussed, but there is question from the Janto Andhra Sakat. Each postbiotic and prebiotic are the same or different?
Yeah, you can
s-
answer that.
Hello.
Yes, sir. Sir, postbiotics and prebiotics are very different as prebiotics are generally sugars which the probiotics utilize for their growth and for their survival in the gut. And they generally feed on that. So during additive therapies and during modulatory therapies, we can give prebiotics. And postbiotics are generally the byproducts of the probiotic. So when they generally ferment the food or ferment in our gut, they produce certain short chain fatty acids like butyrate and all. So these are the postbiotics. And postbiotics are generally chemical, we can say chemicals produced by the probiotics.
Exactly.
And one more question from the, can we explore cow or other herbivorous animal potential gut bacteria as a probiotic formulation for the human consumption? Is it good, safe approach?
Uh, Yes, we can. We have explored in the past also different environment like soil and different other environments to gain probiotics from there. So this can also be I think some paper is there in which they have taken from the ruminants, probiotics from the ruminants. And it has also been used for the production in to increase the milk yield, and in fisheries also we have been using probiotics.
I think that Janto got his answer. And one question to my Anjali, to you, that commercialization of postbiotic or probiotic, which one is easier?
Right now,
But
probiotics is
ultimately,
easier because people are more aware about probiotics than postbiotics. Uh, uh, I think it is more easier now at the present to commercialize probiotics than postbiotics, but we can create awareness about postbiotics also as we will proceed with the research work on postbiotics. Awareness is also should be created among people for the commercialization, because to accept chemicals in this form is quite hard in the layman or common people. So I guess
yes.
awareness is more needed in this field. And as this is very emerging field, so with later or more, as more research will take place, so I think commercialization of postbiotic will also get easier, as we will see more promising results with the studies
Mhm.
with different diseases.
So thank you, Anjali.
Okay, thank you, sir.
Thank you so much. Now the Tejaswani, Manjunath, and Sahana,
Yes,
both of you are working on the
sir.
postbiotic now.
Yeah, yes, sir. I'm working on postbiotic. Especially, I'm targeting the bit rate in colon.
Mhm.
Yeah, you can share your some experience, what you think with reference to the literature and the any information you have.
Okay, sir. As actually, currently I'm working on probiotics on beta carotene availability, wherein I'm seeing a bit rate. It's a postbiotic metabolite, which is having a potential as immunomodulator and intestinal barrier. This thing. Usually, I'm working on DSS, that is colon cancer, clotters, ulcerative clotters. I'm inducing the clotters in the in vitro and in vivo model, and I'm giving this bit rate as to see the mechanism of novelty with the vitamin E homeostasis to increase the mucin production to inhibit the pathogens to enter over there. And also I'm working on intestinal barrier integrity, that is cell to cell junction and tight junction and all those things to prevent the leaky gut in the humans, which is induced in the colon. So basically, bitrate is an energy server in the colon. So when it is incorporated along with the functional foods, it has more beneficial, novel biotherapeutic role compared to the probiotic, because the probiotic is a live microbes wherein we have a challenges of viability and stability over the postbiotics. In postbiotics, it is eat killed or inactivated metabolites which can be incorporated in the functional foods, which has more beneficial mechanism than compared to the probiotics. So I was just working on that.
Thank you, Tejaswani, for sharing your research experience.
Yes.
Sahana is there, so now we'd like to hear from the Sahana.
Can tell what exactly she is doing and what is the result she expect.
She is there or not?
Sahana, are you there? Tejaswini can call her. Meanwhile, we can ask any other participant has any question.
Yes,
Otherwise,
yes,
we can
sure, sir.
close the session. Aditi Joshi is there. She didn't interact, I believe.
She has introduced, Aditi Joshi has introduced herself.
Oh, she is the BSc student, right?
Yes, sir.
She can tell at least whether this session was useful for her to understand.
And also Sari is there, so she can, from the first year MSc. Dr. Vijay Kumar. So there's no more question. Anyone there, if you have any knowledge or your experience or research insight to share, just unmute yourself. Okay. So hello. Dr. Vijay Kumar and then Prashant Marathas are already introduce the service. Okay, so if there are no questions, so now we come to the end of today's session. So thank you everyone for joining today's session, and my special thanks to Prakash Salami sir, Dr. Yaswant sir, Tejaswani, Andhali, Sana, Sairi for joining today's session and making it more interactive and thoughtful discussion were there. So thank you everyone for joining today's session.
Thank you.
Thank you, sir.
Thank you.
Thank you.
Thank
Thank you, Dr. Yaswant.
See you
Nice
again.
of you. Thank you.